So basic mammalian cell culture. Today my presentation, and I will talk about cell culture, application of cell culture, and how you obtain your cell lines, and how you start your own cell line if you need to, and also about first immortal cell line called HeLa. And so before I start, if somebody have you experience with cell culture or not in some labs, because I know that you have experience, and yeah, so you also, yeah, but you saw it, how people do it, yeah, so you will be familiar what I'm talking about. So techniques of cell culture might be described in few statements. It's process of maintaining and keeping cells, maintaining and propagation cells alive outside the organism, or probably another one, it's a tissue culture. It's a method where fragments of cells, of tissues taken, and cells are in tissue disrupted into the cells, and cells are grown in artificial environment and continue to survive and function. And cell culture, one of the method, one of the scientific tools for in molecular biology and cellular biology, providing excellent system where you can study biochemistry of cells and physiology of cells. And it might be studying effect of drugs, effect of cancer biology, and gene therapy, virology. So cell culture has a really broad use in science right now. And we all know that cells is a basic building block of the organism, of the body, and cells form together by morphology and function of cells. Cells are packed, if you can say so, packed together in tissues, and then tissue forms organs, and organs form organs tissue, and then system of organs, they build the whole body, whole organism. And by knowing how function cells on a lower level, on cellular level, we can predict and we can know how cells will behave and how tissues will behave and how organism will survive or behave on different threats or treatments. Yeah, and human body can be divided in 4 different groups of tissue. It's neuronal tissue, nervous tissue represented by brain, spinal cord and nerves. Then epithelial tissue, it's mostly it's lining out your hollow organs and GI tracts. And muscle tissue represented by cardiac, smooth, and skeletal muscle. And connective tissue, it's the biggest part of tissues and connecting all of them in your bones, cartilage, and blood. Under the microscope, you can see how different tissues looks like. So that is epithelial tissue, and we can culture all that kind of tissue. It's epithelial tissue, looks like that, muscle tissue, and neuronal tissues, and mostly biggest part of it's connective tissue. By morphology of cells, cell culture of cells might be divided in groups, and I brought for you the picture that you can see exactly how different tissues and cells from this tissue looks like. So that is epithelial tissue, and it's represented by keratinocytes. And keratinocytes are the cells, they polygonal in shape and grow in discrete patches on a dish. Another, it's fibroblasts. They look a little bit different from keratinocyte, but they are mostly bipolar cells and elongated cells, and looks like that. Another cell class, if you can say so, it's lymphoblastoid cell line cells, and they also represent connective tissue. And of course, myocytes, muscle tissue, and neurons, neuronal tissue. Also, you can classify cells by the way how do they grow on a tissue. It might be adherent cells or anchorage-dependent cells. And anchorage-dependent cells, it's cells, they will be only survived on a dish when they attach to the dish, and they need that attachment to proliferation and growth. And cells, they grow in suspension or anchorage-independent cells. And that kind of cells, they're floating in the media in small clumps or just in a single cell suspension, and they happy this way and continue to proliferate and grow this way. And anchorage-independent cells, it's easy way to handle cells because they are float in a media, as I said, and it's much easier for you to manipulate the cells. So you don't have to think about how you will take it from the dish and like more steps added for adherent cells than for suspension cells. And many labs using cells in their research, and question now where you can get your cell line. And cell lines, we have very nice companies here in US, and it's ATCC, American Type of Cell Culture, and Lonza. It's only two of them, but it's many more. But in these two companies, you can find almost everything that you need for your experiment that you will run. Also, good source of tissues you can obtain from hospitals or medical schools, but you have to find good contact from these people where you can obtain tissue and prepare some paperwork for that, but that is also where you can get some tissues. And scientists are always happy to share with you their cell lines that they're running in their labs, and if you need some cell lines that neighbor labs running, so you can always ask and people will share with you. Or another way, you can establish your cell line by yourself. When first tissue is removed and then tissue is minced in small pieces, and after that we usually in our lab we use digestive enzymes, and digestive enzymes, they works, so for example you have like small pieces of tissues, they're disrupted, and then you add digestive enzyme, and digestive enzyme will release or digest or chew, if you can say so, all small pieces that is like holding cells together. So everything will be disrupted and cells will be coming in suspension as a single cell. One is done, cells are spinned down and put it in a dish, culture dish, in culture media, and they will be growing for 2-3 days, and you will see your cell line is starting. So this way you can establish your cell line. But when you're doing then, that is important to remember about sterile technique and like some equipment and tissue culture dishes or vessels that you have to choose correctly for exactly for your cell line. So it's many of them, and once you will come to that point, you will know what you will need to use. For cell culture condition, it's most cell lines need same conditions. It just depends a little bit different from one cell line to another, but in general, all cell lines need growth media or cell culture media. And cell culture media provides for cells nutrition. It's salt, vitamins, growth factors, and also cell culture media keeps pH of your cell culture at the same level. So it's really important to have also correct cell culture media for you, exactly for your cell line. Temperature, most mammalian and human cells, they grow at 37, 36 degrees. It's optimal temperature for cell lines, but once you will come to that point, you have to check exactly what is your cell lines is better doing at what temperature. Because some, yes? Celsius, yes, it's Celsius, everything is Celsius. It's interesting because all equipment set up all over the world for scientific equipment set up in Celsius. So, and why it's here in US, it's also Celsius, in Europe, it's also Celsius. So it's a standard, so everybody can see what are we talking about. Because if you have in Fahrenheit, you have to convert it in Fahrenheit, but all equipment now in Celsius. And cell culture vessels, dishes, or flasks. Now everything is disposable and made of polystyrene. And once you have your dish, your cells, so you have to put it in some equipment somewhere to where its temperature is kept on the same level, and also gases, as CO2 supply is for cells, it's set up, usually it's 5%. So you have to have a CO2 incubator. And when you're doing if your lab using cells, cell lines, cell culture, so it's a good idea to have cell culture hood. So that is picture of cell culture hood, and cell culture hood provides aseptic environment for your cell culture. So you have to keep everything not sterile, but close to sterile. And in our lab, cell culture hood looks like that, approximately same that what you saw in the previous picture. So that's where we're doing our cell culture. And when you're doing your experiment, you're starting your experiment with cell lines. So first, cells are kept cryopreserved, and it's in liquid nitrogen. And temperature of liquid nitrogen, somewhere minus 145 centigrade. And this way, cells are might be kept 3, 4, 5 years, and they will be viable after you thaw them again. So you take vial of cells, and you thaw it and put it in a media. So I have here flasks that we grow some cells in a flask. And then you do your experiment, whatever you have to do with cells. Your experiment is done, and your excess of cells, you can freeze it again until next time, or you can ask people in the lab if somebody needs it, you can just share with them. But during your experiment, you or scientists, they have to count cells, determine viability of cells, and pathogen cells. So how to do that? In cell culture biology, we have a special term, it's called confluency. And confluency is proportion of surface of your dish where it's taken by cells. So for example, here, so if I will tell you that confluency 30%, it means approximately 1/3 of your dish is taken by cells. So exactly like can you see here, it's 30%, approximately 30% of whole surface is taken by cells. And if I will tell you, so my confluency of my cells in a dish, 90% or 100%, it will be looks like approximately the same. And once your cells reach confluency 100 or 90%, it's a good practice to split them. Because if you will not split cells at the right time, so they will stop to grow, and it's not what you need to do. And how you do your splitting the cell. For example, here the dish, and you have 100% cells covered, surface covered. So it means you have to split them. So you disrupt cells, dislodge from the dish, and you made 2 dishes or 3 dishes or 5 dishes. It depends on your cells. And how we do that. So here's small cartoon how cells looks like on that picture. And now question how you will take them off of the dish. So it's two ways how you can take them off. First, it's a mechanical method. So you can scrape them with a spatula, but it doesn't matter how gentle you are, you can damage yourself or even kill it during disruption. And many people use chemical methods, and chemical methods we use enzyme called trypsin. And trypsin will cut away. So you see, cells are attached to the surface. So small molecules here, they holding cells on a spot to the dish, and they also hold each other. So, and trypsin, when trypsin will come, so trypsin will cut all that molecules, and cells will be floating. So here trypsin come, and all molecules disrupted, and they start to float. Once they floating, and you can take small, so at that point, once they're floating and round, so you will take your cell suspension and divide it in 3, 4, 5 dishes. It depends on your cell line, how do they like to be, like what proportions they like to grow at. And once cells are place it in a new place, in a new dish, it's coming more space for them and new nutrition, and cells in probably 2-3 hours enough, and they will be attached again to the surface and start to grow again and proliferate. That is all picture how it looks like, and all like schematic picture. And I would like to show you how it looks like under microscope when you're doing exactly your trypsinization. So pay attention how cells will be, like now they're spread and sitting and fine, but now they will be shrinking. Yep. So they became round, and you can split your cell line right now. So once you split in your cell line, it's a right time to count cells and find out viability of cells. And to do that, you have special device called hemocytometer. And hemocytometer, it's made of glass and have 2 grids on top and bottom side. Yeah, and where you load your cells from bottom and from top. And in this way, and it's made of glass. I don't know if I said or not. So it's made of glass. And you will see exactly the same picture under the microscope. So that is a microscope, and exactly what you see under microscope. It's a grid made of 9 bigger squares, and each 9 of squares divided into 16 smaller squares. When person would like to find out cell concentration, you have to count 1, 2, 3, or 4 squares, and then you will make average of it, and you will find the concentration of your cells. I just would like to show you hemocytometer that is now on the market, and it's disposable hemocytometer. Before it was glass one, and you have to clean it after each time you count cells. But it is disposable, so you count and you just throw it away, and it's done. So how to count cells? So that here, hemocytometer with grid, and you have to count your 1, 2, 3, or 3 or 4 squares usually. And green cells, green dots, it represent live cells, and the blue dots, they represent dead cells. So, and all that cell culture is stained with trypan blue. Trypan blue is special stain that enters dead cells much faster than alive cells. So once you count, stain your cells with trypan blue under microscope, you can determine dead cells and alive cells. So here, one of the 9 squares. And it's quite clear that you can count, so that you will count cells all inside that square. But sometimes it will be questionable for you, should I count cells that sitting on the edges or not, or how I deal with it. So, so I propose for you that you count, and in our lab we count also this way. So we count all left side and top side of the slide. Whatever slide it's touching outside, it's touching inside, or sitting exactly on the grid, we count all of the cells. But we don't count any cell that doing the same, but on a bottom or right side. Bottom or right side. So in this way, it's kind of equal. Of course, it will be not exactly equal, but some kind of equal. So you just cut some cells, not wanted cells. So, and here's small presentation and all like summarization, summary how to count cells in a lab.
A hemocytometer is a device that is used for counting cells. It's a modified microscope slide containing 2 identical wells or chambers into which a small volume of cell suspension is pipetted. We have already removed 100 microliters of our cell suspension and placed it in a microcentrifuge tube. Dilute the suspension by adding 100 microliters of trypan blue. Trypan blue is a dye that helps us distinguish between living and dead cells. The dye passes through the membranes of dead cells, so they will appear blue under a microscope. Living cells exclude and will appear mostly clear. Load both chambers by pipetting the suspension under the cover slip. Now place the hemocytometer under the microscope. Each chamber is divided into a grid pattern consisting of 9 large squares. Each square has the same dimensions and contains 10 to the -4th power milliliters of suspension. The rules for counting cells sometimes differ from lab to lab. In our lab, we count cells in the 4 large corner squares and the center square. Let's count the cells in the first square. 1, 2, 3, 4, 5, 6, 7, 8. So what about the cells that are touching the outside boundaries of the square? In our lab, we count the cells that touch the top and left boundaries, and we ignore the cells that touch the right and bottom boundaries. 9, 10. We need to count the number of both living and dead cells. Remember, the dead cells will appear blue. Occasionally, you will see artifacts, objects or debris that appear blurry and don't have a well-defined shape. This is an example of an artifact. We won't include it in our count. Proper storage, cleaning, and handling of the hemocytometer will minimize the number of artifacts. There are 10 viable cells and 1 non-viable cell in the first square. Now, the top right square. There are 9 viable cells and no non-viable cells. Next, let's count the bottom right square. There are 11 viable cells and no non-viable cells. And now the bottom left square. There are 10 viable cells and 2 non-viable cells. And finally, let's count the cells in the center square. Sometimes cells will appear as clumps or small groups. It may be difficult to determine exactly how many cells are in a group. The method of counting clumps of cells differs from lab to lab, so be sure to follow the procedure in your lab. We will count this clump as 2 cells. There are 14 viable cells and no non-viable cells in the center square. The total number of viable or living cells from all 5 squares is 54. The total number of non-viable cells is 3. Now that we have counted our cells, there are several calculations we need to perform. First, let's calculate the percentage of viable cells. Here's the formula. 54 viable cells divided by 57, the total number of cells, gives us 0.947. Multiply by 100 and the percentage of viable cells is 94.7%. Next, let's determine the average number of cells per square. We counted 54 viable cells. We divide 54 by 5 because we counted in 5 squares. The average number of cells per square is 10.8 cells. Now let's calculate the dilution factor. The dilution equals the final volume divided by the volume of cells. Our final volume is 200 microliters because we started with 100 microliters of cells and added another 100 microliters of trypan blue. 200 divided by 100 is 2. Therefore, the dilution factor is 2. Next, we need to calculate the concentration of viable cells, the number of living cells per milliliter. Our average count per square is 10.8. The dilution factor is 2. 10.8 times 2 times 10 to the 4th power equals 216,000 cells per milliliter. We can write the concentration using scientific notation as 2.16 times 10 to the 5th power cells per milliliter. From our calculations, we now know the concentration of cells in our culture is 216,000 cells per milliliter, and approximately 94.7% are viable living cells.
So now here we have a cell line called HeLa, and before I start to show you another piece, so what do you think, I just would like to ask you, what is your guess? Confluency will be here on this side. What is confluency here? Just guess. It's no exactly number is written, just how you feel. Yeah, for this side. Yeah, probably, yeah, 30%. It's all how you feel about. So it's not like guess, it's not I will tell you, no. It's not 20, it's 25. It's just how you feel. And on this side, on another side, what do you think concentration here or confluency is here? 70 to 80. Yeah, 70 to 80, yeah. They're quite full. The dish here is quite full. Yeah. So it's how people speak in a lab. So, for example, you have an experiment, and I'm asking you, so do you have your experiment today or not? And you tell me, no, I don't have, because my cells are really low confluency, and it's no way that I will have enough cells today. Or another day I will ask you, do you have experiment today? If you're doing your experiment today, you say, yes, I have enough cells today, they're about 80 or 90% confluency, and it will be enough for my experiment. So it's why we have that term confluency, so it's really useful in experimental design. And now I will show you and tell about HeLa cells. HeLa cells, it's a first immortal cell line. It's and so first cancerous immortal cell lines. And a lot of labs and almost everybody in scientific world, they know what is it. And I would like to show you small also movie about that HeLa.
Imagine something small enough to float on a particle of dust that holds the keys to understanding cancer, virology, and genetics. Luckily for us, such a thing exists in the form of trillions upon trillions of human lab-grown cells called HeLa. Let's take a step back for a second. Scientists grow human cells in the lab to study how they function, understand how diseases develop, and test new treatments without endangering patients. To make sure that they can repeat these experiments over and over and compare the results with other scientists, they need huge populations of identical cells that can duplicate themselves faithfully for years. But until 1951, all human cell lines that researchers tried to grow had died after a few days. Then a Johns Hopkins scientist named George Guy received a sample of a strange-looking tumor. Dark purple, shiny, jelly-like. This sample was special. Some of its cells just kept dividing and dividing and dividing. When individual cells died, generations of copies took their place and thrived. The result was an endless source of identical cells that's still around today, the very first immortal human cell line. Guy labeled it HeLa after the patient with the unusual tumor, Henrietta Lacks. Born on a tobacco farm in Virginia, she lived in Baltimore with her husband and 5 children. She died of aggressive cervical cancer a few months after her tumor's cells were harvested, and she never knew about them. So what's so special about the cells from Henrietta Lacks that lets them survive when other cell lines die? The short answer is, we don't entirely know. Normal human cells have built-in control mechanisms. They can divide about 50 times before they self-destruct in a process called apoptosis. This prevents the propagation of genetic errors that creep in after repeated rounds of division. But cancer cells ignore these signals, dividing indefinitely and crowding out normal cells. Still, most cell lines eventually die off, especially outside the human body. Not HeLa, though, and that's the part we can't yet explain. Regardless, when Dr. Guy realized he had the first immortal line of human cells, he sent samples to labs all over the world. Soon the world's first cell production facility was churning out 6 trillion HeLa cells a week. And scientists put them to work, in an ethically problematic way, building careers and fortunes off of Henrietta's cells without her or her family's consent, or even knowledge until decades later. The polio epidemic was at its peak in the early 50s. HeLa cells, which easily took up and replicated the virus, allowed Jonas Salk to test his vaccine. They've been used to study diseases including measles, mumps, HIV, and Ebola. We know that human cells have 46 chromosomes because a scientist working with HeLa discovered a chemical that makes chromosomes visible. HeLa cells themselves actually have around 80 highly mutated chromosomes. HeLa cells were the first to be cloned. They've traveled to outer space. Telomerase, an enzyme that helps cancer cells evade destruction by repairing their DNA, was discovered first in HeLa cells. In an interesting turn of fate, thanks to HeLa, we know that cervical cancer can be caused by a virus called HPV, and now there's a vaccine. HeLa-fueled discoveries have filled thousands of scientific papers, and that number is probably even higher than anyone knows. HeLa cells are so resilient that they can travel on almost any surface. A lab worker's hand, a piece of dust, invading cultures of other cells and taking over like weeds. Countless cures, patents, and discoveries, all made thanks to Henrietta Lacks.
And in 2010, its book was published, The Immortal Life of Henrietta Lacks. And I just recommend this book for you. Just take a look. You will not only find out how this woman was lived and all the circumstances of that cell line, how it was made, but also it's very interesting to see pictures over there and how cell culture was done 60 years ago. So people was doing it not like nice, real cute dishes like we're doing right now. They was doing cell culture in the buckets. So, and no sterility, no nothing. And it's just really amazing how it was done just 60, 50 years ago. So that is my recommendation for you, just to find out this book and just to check it out. And that is stained HeLa cells. Yeah, that's all for today. Thank you.